Hello, everyone. Welcome, and thank you for joining the 25th webinar in our Pandemics and Society series. Today's webinar topic is Mirror Biology, Global Risks, National Security Concerns, and Practical Actions. I'll now pass it to our moderator, Wilmot James, Senior Advisor to the Pandemic Center and Professor of the Practice of Health Services, Policy and Practice at the Brown University School of Public Health, to begin today's conversation. Wilmot?
Hello all. Good morning, good afternoon, good evening, colleagues and friends, and welcome to our discussion today. Last year, I was honored to join a group of scientists, including 2 Nobel Laureates and 16 members of national academies from around the world in publishing a warning against the building of a technology that some of them had spent years working towards. We describe it as a mirror bacterium. So mirror bacteria are hypothetical synthetic organisms built from mirror image forms of proteins, amino acids, DNA, and other biomolecules used by life on Earth. In an analysis published in Science, our group argued that such organisms could be built within the next 10 to 30 years and could pose an extraordinary threat if they were. Our analysis is that mirror bacteria could be resistant to many mechanisms of immunity in humans, in non-human animals, as well as possibly plants. They could also be resistant to the predators that keep populations of wild bacteria in check. It's plausible then that mirror bacteria could act as an invasive species, causing fatal infections as they spread and irreversibly disrupting ecosystems in the process. Scientists cannot yet make mirror image versions of all the components that would be needed to create a mirror bacterium, and no researcher has successfully booted up a normal bacterium from entirely non-living parts. And so the threat, my friends, is not imminent. The questions raised by mirror biology, however, are pressing and need not just consideration, but practical action. To discuss the risks and challenges posed by mirror biology to global health, national security, and international collaboration, and to illuminate what practical steps can be taken to stop those risks from developing, I'm very excited to be joined by 2 experts in the field. I'd like to first welcome Dr. James Smith, an adjunct associate professor at the J. Craig Fenter Institute and the deputy director of the Mirror Biology Dialogues Fund. James was also a co-author of the mirror bacteria analysis published in Science last year. To lay the context better and to better understand the risks, Dr. Smith will spend a few minutes presenting some slides. He will also present a readout from the first international conference on mirror biology that was recently held at the Institut Pasteur in Paris. I would also like to introduce my colleague, Dr. Beth Cameron, who is a leader in national and global biosecurity and has worked at the highest levels within and outside of government. Beth spent 2 tours as a special assistant to the president on the White House National Security Council, establishing and leading the Directorate of Global Health Security and Biodefense under 3 presidents. She's also a non-resident senior advisor to the Center for Strategic and International Studies, Global Health Policy Center, a life member of the Council on Foreign Relations, and a practitioner senior fellow of the UVA Miller Center. She's also a senior advisor to the Pandemic Center. So thank you to both of you, James and Beth, for joining us today. So over to you, Dr. Smith, for a short presentation, which will be followed by a panel discussion.
Great. Hopefully the slides are looking okay. Perfect.
Yes.
Um, uh, Perfect. So thank you, Wilmot. Wilmot already gave a really good overview of the risks here, but just to recap some of the key points, what we're talking about today are the risks of creating synthetic mirror life. What that means is a life form that would be made out of molecules that are reversed in terms of their chirality compared to what happens in nature typically. So if you look at this figure on the right-hand side, all life is composed of molecules like amino acids and DNA that can exist in one of two different mirror image configurations, just like left and right hands. And this property of handedness is what we mean by the word chirality. All life on Earth uses biological molecules of a consistent chirality, and that basically allows them to fit together and to interoperate. But those molecules can be constructed synthetically in the lab in their mirror image versions. So normally DNA, for example, rotates only to the right, which is also the case for Fuseli pasta, if you've ever looked at that. But it can be made so that it will also rotate so that it would rotate to the left instead. And according to research that we published last year, we expect that in roughly the next 10 to 30 years, it could be possible to assemble these reverse chirality molecules into an entire self-replicating mirror-bacterium. And a mirror-bacterium would be the simplest viable form of mirror life that we could realistically create. So in isolation, a mirror-bacterium would behave identically to a normal bacterium. All the molecular parts would be exactly the same. They would just be mirror images of normal parts. And we know from physics that they would behave in the same way. And there doesn't appear to be very strong practical benefits to creating them. I think the primary motivation for pursuing this work in the past has been that it's kind of a grand, interesting challenge of science, although there are some potential applications for biomanufacturing. And the risks of mirror life have been discussed occasionally in the literature or alluded to in the literature as early as the 1990s. But the first detailed analysis was done late last year, and this was the one that where Wilmot and I were both co-authors on. Published this paper in Science in December, along with a 300-page technical report, which went into much more detail around a lot of the questions here. So what actually are the risks? The key point is that mirror could plausibly pose an unprecedented risk to humans, to animals, to plants, and ecosystems. And I think there are two key points to understand here. One is that they potentially can evade immunity across a very wide range of multicellular species. And the second is that they can pose ecological risks because they would be able to escape many of the typical biological controls that keep bacterial populations in check. Fundamentally, our immune systems rely on molecular interactions that would not work with mirror because the chirality essentially wouldn't match, kind of in the same way that a left hand can't fit into a right-handed glove. And the result of this, in many different places in the immune system, these interactions not working as they should, would be a dramatically weakened immune response. It seems very plausible that this would lead to fatal infections in humans and many other species too. You might be thinking, how could a mirror survive in nature if it requires these chiral interactions? What would it eat? But it turns out that mirror could very likely survive on achiral food sources that don't have mirror images. And we know this from natural chirality bacteria. We know that E. coli, for example, can live on a variety of achiral food sources. So we think it's likely they'd be able to find sufficient nutrients. If they can find sufficient nutrients to grow, then the question is, how quickly are they killed? Normally, bacterial populations are killed by viruses or predators, and we know for sure that these viruses would not work with mirror and it's pretty likely that the other forms of predation would be substantially reduced too. So if a mirror released into the environment, it seems like it could grow exponentially, potentially invading a large fraction of multicellular life on Earth. You might ask, could we contain mirror in the lab? And I think, unfortunately, it seems very difficult to do this in a way that would be reliable. All that it would take is one potential mistake here, one escape from the lab for catastrophic damage to be caused. And I think more importantly, it's very difficult to have sufficient confidence that a malicious actor would not be able to undo safeguards that were in place here. The bottom line of the work that we put out was that we didn't think mirror should be created, and some of the enabling work that would allow one to create mirror should not be pursued. This was put forward as a provisional recommendation in the science paper. Since that paper was put out, there have been a series of meetings to think about these risks in more detail and discuss what should be done about them. And I'll just point that there have been several convenings. I'll just point to two that I have personally been involved in. The first was at the Spirit of Asilomar and Future of Biotechnology Summit, which happened in February and included an international group of ethicists, policymakers, industry, and others. The result of this meeting was a statement signed by nearly 100 participants saying that mirror bacteria should not be created and that we need to figure out what governance should look like. More recently, as Wilmot mentioned, there was a meeting in Paris on the risks of mirror life, which was the first international meeting specifically dedicated to this topic. And similarly, I think there was very broad agreement that mirror life should not be created. And so we need to move forward and think about how can we ensure that it will not be created. As we look ahead, there are many open questions still. Meetings like this, I think, are going to play a critical role in determining and thinking through what we should do. People in the audience would love to hear thoughts from them, but there will also be a series of other meetings happening throughout 2025 and 2026 to continue the discussion and move towards what exactly should we put in place to ensure that mirror bacteria can't be created in the long term. So I'll stop there and hand back over to Wilmot.
Many thanks, James, for that overview and also just walking us through some of the basic elements of science that can frame the understanding of the risks. I would like to start off by asking you a question just to follow up on your presentation. And the question's about mirror life versus mirror biology and the technologies that could enable mirror life, but do not actually create mirror life itself. So it's a distinction about enabling a process versus the actual creation of mirror life itself. Can you explain the difference? It's a critical distinction.
Yeah, definitely. This is, I think, a very important question. The analysis that I talked about and the risks that I talked about are very specifically focused on mirror life itself. This would be a self-replicating organism. But there are other technologies that together, when taken together, might help you to create mirror life, but on their own do not present any of these special risks. So the pathway through which mirror life is most likely to be created, in my view, if it were to be created, is through a combination of creating mirror biomolecules and research into natural chirality synthetic cells. Two things need to be true to be able to make a mirror cell. You need to be able to make all of the components of that cell, and you need to be able to put them together and create life out of essentially inert material. The mirror molecules that you would need to create mirror life, some of those would be very complex and are out of reach in terms of what we can do at the moment. But some of them, simple proteins and nucleic acids, are already used in the lab or explored for therapeutics and have, I think, promising applications. And so in the paper and in the discussions since the paper, I think it's been very clear that no one is proposing any sort of restrictions on these mirror molecules that have the therapeutic benefits, have scientific benefits. And the thing that's concerning is the full mirror cell. There are technologies kind of in between those where there's more of a question mark in terms of where do we draw the line. I think in the long term, for us to be able to ensure that mirror life can't be created, it's going to be necessary to draw lines, not just at the very final step, but somewhere on the pathway to creating mirror life. But where we should draw those, I think, is still an open question and something that a lot more research and thinking will be needed on.
Thanks very much, James. The whole question of benefits when it comes to the manufacture of or assembly of biomolecules is something we'll return to discussing. I mean, the conclusion has been that the risks outweigh any benefits by far along the road, but we need to discuss what those benefits are for those single molecules that are not replicating, obviously. But let me first of all turn to Beth to pose a question. So you heard James, he spoke about some of the risks associated with mirror life. Can you talk for a moment about the frameworks that governments have used to address other risks associated with the life sciences? What kinds of next steps or frameworks could be considered in this particular case, given the fact that we are categorizing this as a risk in a class of its own, something we've just never seen before? So is it different or similar from other kinds of technology-related risks? In other words, are there models out there that we can use, we can look towards using to build a framework that is robust?
Thanks. And thanks for having me on today. This is a really, really important topic. And maybe if I could, I want to just start by saying that this is a really rare instance when we have the chance to think about policy and governance and practice before the horses left the barn. So much more frequently, I think we in this community and many of our listeners find ourselves in the position of being reactive. So when the 1918 flu virus was synthesized, that led us to put in place gene synthesis screening practices. When the anthrax attacks happened in the United States, that led us to put in place our select agent rule, our practice, our policies for overseeing materials that might have the ability to cause harm in biology. When there was gene editing announced in twins in China, that sparked a flurry of activity around safety and security with gene editing. When the H5N1 experiments happened in 2012 in the United States and in Netherlands, that sparked over a decade of work on dual-use research of concern policies. And so I say this both by way of saying we have a chance to get ahead of these of mirror life, which has not been created and might be as much as 10 to 30 years on the horizon, but also to give a sense of the types of policies that are in place for different kinds of technologies. I think in the case of mirror life, we're talking about something where the risks so far outweigh any potential benefits that we already have a slew of scientists, as James has mentioned, saying we already need to put in place at least a pause or a moratorium on that work, on mirror life itself. But maybe I'll talk just a little bit about some of the things we might be able to do in that time frame window. That valuable window of time, I think, is 10 to 30 years, but we've also been, I think, historically really bad, frankly, in the biotech community of understanding how fast technologies can move. So I think we might see some shifts, and we should pay attention to those enabling technologies that James just mentioned that might shift that timeline and make it shorter. But what I think it means is that right now we really need to engage a broad group of stakeholders in the scientific community and around the world. We need to be talking to policymakers so that they understand what mirror life is versus what mirror biology is, and so that we do have a better understanding of what the enabling technologies are and where to draw lines on our ability to create mirror life. And if you ask, what kinds of frameworks we might kind of consider, maybe I'll just put a few on the table so that we can get a discussion going and maybe stimulate some great questions and answers from this awesome audience. So I think one is a conversation with research funders. So I think first and foremost, engaging with people that fund biomedical research, they have a really a big bully pulpit, an opportunity to say, we don't think mirror life makes sense. And so we are going to work with the scientific community to understand at what point we will not fund research that is so far on the pathway to developing mirror life. And I think even before that, saying publicly, we don't think mirror life should be created. So that is not so much a framework as setting a norm in the system. That kind of norm to get there, I think, really does need broad engagement with funders, with researchers, and also with private sector stakeholders, with ethical, legal, and social scholars, and the like. And there's been a lot of work to get to that point, and James outlined some more work that's going on, but really welcome views from our listeners about where else that engagement and that discussion should happen. Second, I think normative statements and policies, like I mentioned, are going to be really important. And those happen, I think, first in the funder and scientific community, but also can be discussed in other important bodies like the World Health Assembly, other UN bodies. And I think in the United Nations, having a broad understanding of where mirror life could play a role, I think is important, because we're talking not only about an impact on human health, but a really important impact on agriculture, as Wilmot mentioned at the beginning. Third, I think that we need to be talking to innovators, people that are developing technologies that will make it easier to create some of the enabling tools like synthetic cells or mirror ribosomes, so that we can be thinking about what kinds of guardrails could actually be built in for overseeing this kind of work, making sure that we're not enabling the development of mirror life, but we are creating the opportunities in life sciences that we need to continue to create going forward in mirror biology, for example. And then fourth, I think we do need to look at the policies that we have in place. I think when we look at policies like dual use research of concern policies, we have some guidelines for practices that we can use in, biosafety committees, oversight bodies, research funding oversight. But I think we also have to keep in mind that those policies were created to look at research that has both a benefit and a risk, and to weigh those benefits and those risks. And I think in the case of mirror life, we're looking at a situation where the actual creation of mirror life may not have any benefits at all. So using those practices to understand the enabling technologies and to ensure we're not creating mirror life, but recognizing that the lexicon that we're using is a little bit different when we talk about mirror biology. And I think that makes it really important to continue to have these conversations. And then I think last but not least, we have some international institutions that look at dual use research and emerging biotechnology that could be really important to leverage. The first one that comes to my mind is IBIS, the International Biosecurity and Biosafety Initiative for Science, which brings together private sector institutions and technology developers and looks at things like the ability to screen DNA synthesis orders. There may be a role for these existing platforms to be more engaged in the mirror life conversation, and to be able to understand if there are best practices in developing the enabling technologies and understanding where to draw the lines, that these institutions that already exist are involved in that conversation. So maybe I'll just stop there and go back to you, Wilmot, but I think this is a really huge task and one that we really don't have time to waste here.
So thank you very much, Beth. You referred to having a compelling conversation with funders, because without funding, this will not happen, right? And so there are a number of potential sources of funding. There's public finance around this through the science community, but there's also corporate money that goes in, that is driving some of the research in a sort of the precursor molecular investigations when it comes to developing therapies. So I'm turning to James. Could you share with us what kind of funding we're talking about here in terms of what it would take to get to the point where you would have sufficient investment to, in fact, build a mirror-bacterium, and what the strategy would be to deal with not only public finance, but also corporate investments into this area?
Absolutely. So I think the the timeline that I referred to of something like 10 to 30 years for it being possible to create mirror life. As Beth pointed to, we're not very confident in this, and it could be shorter. So another way to think about how far away we are from creating mirror life, I think, is how much would it cost? How much would a dedicated effort need to spend to get there? The number that many synthetic biologists, or the range that many synthetic biologists end up in when this question is posed, is something like $500 million to $1 billion in order to be able to create a mirror cell. So this is a substantial amount of money, but it's not an absurdly large amount of money. And a really dedicated effort to do this could bring timelines for creating mirror bacteria further forward. Fortunately, at the moment, I don't think there's anyone who is specifically trying to create mirror life. Before the publication of the science paper, there had been grants aimed at booting up mirror synthetic cells, but the principal investigators of those grants were in fact on the paper calling attention to these risks and pointing out that they didn't think mirror life should be created. The question of industry, I think, is a really important and interesting one. There is investment in mirror molecules as therapeutics, and there are a small handful of companies that are investigating mirror therapeutics, including in clinical trials. And there are, in fact, I think, 2 FDA-approved drugs that are mostly made up of mirror amino acids. So this is an area of industry that's already somewhat established. But there is no industry investment and no industry interest, as far as I can tell, in creating mirror cells themselves. So I've spoken with, at least as far as I know, all of the people at these, people from all of the companies that are developing mirror therapeutics, and they don't think it will be necessary to make mirror life in order to manufacture their products. And in fact, given that the products are already in some cases in quite late stage clinical testing, we can be quite sure, I think, that that's not necessary. You can make these molecules through other approaches.
So
I'll
thanks.
stop there, Wilmot.
Yes, if you could just elaborate on one thing, I'll turn to Beth as well to respond. But could you share with the audience what the therapeutic benefits are of producing single molecule based mirror therapies?
Yeah, absolutely. The key properties that make mirror biomolecules of interest are that they are resistant to degradation by proteases. So the enzymes that normally break down proteins in the blood just don't work with mirror therapeutics for the same reason that the many aspects of the immune system are likely to be impaired. They don't fit together properly like hands in a glove. And this means that they also have lower immunogenicity, so they're less likely to provoke an unwanted immune response when patients are dosed with them. Those are the 2 key reasons I think that there's interest in exploring mirror therapeutics.
Okay, no, thank you very much. So over to you, Beth. Any reflections on how one goes about, let's call it, choking the funding? So how does one go about it in terms of process? And also being able to, at the same time, think about how you deal with the dual use aspects of ongoing research, for example, when it comes to single molecule work.
Yeah, I think this is a critical point because there is a lot of ongoing work with mirror biology, as James just outlined, and there is concern about stopping that work, which is not what the people working in this field who are arguing for a pause on the creation of mirror life are arguing for. And so what that means is that there needs to be a really detailed conversation at the scientific level about where the lines should be drawn. And that conversation is ongoing. I don't have an answer to that question because there are a number of ongoing discussions happening amongst the people who are actually doing this work to understand at what point the benefits either don't exist anymore or the risks so far outweigh the benefits because we're so far down the road to mirror life if a specific technology along the road can be created. And this is, we have not yet set those lines. And James, I think I welcome your views on where the scientists are sort of trending these conversations. I know you're getting ready to hold the discussion that's going to happen, I think, in Manchester later this year to really dig into these issues from a very technical level. But as we get closer to being able to develop a synthetic cell that is not mirror form, but a regular synthetic cell, as we get closer to be able to develop a ribosome that is mirror form, we will be much closer, I think, to the pathway of being able to develop a synthetic cell that is entirely mirror. And so the question is, at what point along the way should the line be drawn? So, Wilma, that's a long way of answering your question to say, first, we have to figure out where the lines are. And the scientists really do need to be driving that conversation. And I think the scientists are driving that conversation. That doesn't mean the policymakers shouldn't be part of the discussion. The policymakers need to be part and parcel to this so that they can be looking at these technologies and saying, okay, presuming we set a line in a certain place, how do we understand if we get a funding proposal or if we're putting in place a governance pathway, how will we determine that that is what we would not fund? What are the triggers for that? One of the things that came up recently in a conversation I had the privilege of being part of in Amsterdam last week that was put together by the Nuclear Threat Initiative, there was a lot of discussion about whether we need a global body, a global experts group that is geographically representative, consists of scientists that are working on these technologies for therapeutic benefit, mirror biology, and includes people that are looking at both the agricultural risks and the human risks associated with mirror life, to actually have a body that would be the body that would call for a pause or a moratorium on funding mirror life, but at the same time would revisit the tools and technologies that would be enabling so that we could set triggers for policy development, and also to revisit as we learn more and more about these tools and technologies. I think that's a really great strategy that we call for a funding pause, but at the same time put scientists together regularly to review what they know and what they don't know so that it's very transparent, so that there's a broad understanding of what's happening with the technologies that would enable the development of mirror life. And so we can have a real conversation about things like detection. How would we know if mirror life is being created or if there are laboratories developing it that we don't know about? And the idea here is to be transparent so that we know all of the people who are developing this work and so that they're actually part of the conversation. And I really think that so far in this conversation, that has happened incredibly well. I think that's setting us on a path for success with something that frankly we haven't been able to do in the past, which is draw out a place where we should be not conducting research before we get to the point where it's possible to do that. Usually we see something happen and then, as I mentioned earlier, we're incredibly reactive, and it's very hard to stop something this catastrophic once it is out.
Thanks. So back to you, James. So could you just share what the plans are for how one determines on a scientific basis where that red line is to be drawn? The plans for the Manchester meeting and beyond that. And while you're addressing that particular question, also what the strategy is on the part of this effort to engage, sort of vigorously engage the entire world, and not just the North and not just a selection of countries, but given the risks we're facing, we need to engage globally the diversity of audiences.
Yeah, absolutely. So maybe just to briefly follow up on the question of where lines should be drawn and the role of funders here. I think the thing that there's very clear agreement on is that the final step of creating mirror life should not be done. And so I think a near-term action that funders can take is to commit and publicly commit to not funding that final step of creating mirror life and not funding research that specifically is aiming to do that. If we move further back from that final step, I think the question is much more complicated. In the science paper, we put forward two potential lines. One was a mirror genome and one was a mirror proteome that everyone, at least on that paper, agreed would not have significant value to create on its own and would be taking us quite close to being able to create mirror life. If we look further back than that, I think there are really tough open questions. And how are we going to decide what to do there? I think a critical input is a better understanding of what are the benefits of these potential technologies that in different places where the line could be drawn. As I mentioned, there are two areas of research, kind of mirror biochemistry, you might call it, and synthetic cells, that I think the convergence of those could lead to the ability to create mirror life. But both of those have potential benefits at different points. And I think to go from where we are today to a determination on where that line should be drawn, we need better technical information. We need to understand what are the benefits of these technologies and how close do they get us to being able to create mirror life. And there's a meeting at the University of Manchester that a number of synthetic biologists with the most relevant expertise, or some of the most relevant expertise in those specific technologies, will be there to think about that. But I don't think this decision can be made only by scientists. This is a risk that potentially affects everyone globally, potentially affects a very wide range of different ecosystems, different species. And so it can't just be a small group of synthetic biologists who make this decision. And so I think it's critically important that people with broader expertise, so policymakers, ethicists, other social scientists, immunologists, ecologists, are involved in thinking about what is the right place here to draw the line. And I think that goes beyond just scientific expertise. I think international collaboration here is absolutely critical, to Wilmot's point. This is probably the leading research that would be most relevant to creating mirror life is happening in the United States, in China, and in Europe. But I think everyone would be affected by the impacts of this and therefore needs to have a say in the conversation. And so there's been a great deal of international collaboration so far in this effort. Many Chinese scientists, American scientists, people from South America, India, and I think really all over the world. I think on the Asilomar statement, there were people from all 6 inhabited continents who signed on as supporting that this should not be done. And it's going to be really important to keep that up going forward and ensure that all of the people that have a say in the outcomes here also get to decide where this line should be drawn and what else we should do to mitigate the potential risks.
And Wilma, maybe I'll just jump in also to say that when I mentioned that I think scientists should be driving the conversation, I think they should be driving the conversations about the technical benefits and risks. I think that this does need to be a much broader conversation. And I do think one of the things I'm hearing consistently from scientists is that having scientists be able, hopefully when a pause or moratorium is put in place, to be able to provide evidence that impacts on that decision, I think allows for a conversation to continue that reinforces that norm and brings the scientific evidence to bear on any potential benefits that might be discovered about enabling technologies along the way. So I do think having that strong scientific, global, geographically representative group of experts in synthetic biology and immunobiology and biochemistry, protein chemistry is really important to keep going while we're also having these really important policy conversations. And that means engaging inter-academy experts, scientists within the inter-academy panel. Our own National Academies of Science is launching a workshop this fall to talk about this issue, which I think is positive and should be global engagement. We need to engage within the UN system, including in areas that talk about emerging technology and biotechnology. That's everything from the UN committees that look at life sciences more broadly, to the Biological Weapons Convention, where mere life is not a weapon unless somebody intentionally were to use it to cause harm. But there's a lot of conversation about emerging tech and tools. And so educating people and policymakers in that context, I think, is also continually really important.
So I want to go back to a fundamental science question before I pursue the issue around deliberate use and deliberate manufacture. Beth, you're raising that particular issue. But one of the questions in the chat from one of our participants goes as follows: is the experimental evidence for the immune evasion claim? As in, are there studies evaluating the immune response to mirror proteins, for example? If you inject mirror proteins into an immunocompetent mammal, do they mount an immune response? Either Beth or James.
Yeah, I'm happy to jump in on this one. Maybe just to say up front, something that I didn't make clear in my initial presentation is that when I first heard about this issue, and I think when many of the scientists, probably all of them that I've spoken to about this, first heard about it, I was very skeptical that the risks could be as bad as they appear to be. I think this is a very extraordinary claim here, and so it deserves serious scrutiny. And every one of the scientists that was on this science paper was initially sort of tasked with trying to find what's the hole in this argument, what's missing here, and ultimately we couldn't think of an issue that kind of ruled out this risk. But it's certainly something that initially I think is very hard to hard to believe. But there is, I think, actually very strong experimental data for a number of the claims. Of course, it's indirect. We don't have mirror life to be 100% sure. And to the specific question asked, yes, there is experimental data in which mice get injected with mirror proteins, and the antibody responses are much weaker than when injected with non-mirror proteins. There's also a lot of other relevant experimental data that suggests that the immune response will be impaired. So, for example, mirror proteins do not get cleaved by natural chirality enzymes, including those that break down proteins into peptides for antigen presentation. So we know that doesn't work properly, and this interrupts T-cell response. We know that mirror antigens are poorly recognized by T cells, even if they do end up being presented as experimental data from this. And then we can also look at human immunodeficiencies. So if you look at something like MHC class 2 deficiency, and this is just one part of the immune system failing to work, we know what happens to people that have this. Generally, they die in childhood. And with mirror bacteria, we're not talking about just one deficiency. We're talking about deficiencies throughout many different parts of the immune system. So I think, unfortunately, there's quite compelling evidence that that the immune risks would be very severe.
Thanks very much, James. If I could now return to the BWC, Beth, and so you brought up the Biological Weapons Convention. Anybody who deliberately assembles a mirror bacterium is in violation of the BWC because it's a weapon of, it would be a weapon of note. So in your estimation, how I'm trying to think of the right word. I mean, how robust would the BWC be in terms of setting not only a series of guidelines, but actually making it possible to stop a rogue state, a rogue actor from finding the right amount of money to put behind this in order to make it? I mean, what's the protection here?
Well, I'm interested in James's view too, because there's been a lot of debate around the BWC and how much mirror life should be featured in discussions about how much the BWC should be featured in discussions about mirror life. And the reason for that is because, in my opinion, Wilmot, you're right, if someone were to assemble a mirror bacteria and there's no other purpose for it, then you could by inference that the intent is to use it to cause harm, because we know what the harms would be. At the same time, there is that piece of the BWC intent. And so at the moment, we don't have a mirror life. It hasn't been created as far as we know. And we have a consensus that creating it would actually cause massive consequences. And so I think the BWC is a relevant and important norm for discussing mirror life. In other words, having statements and discussions about what you said, I think very matter-of-factly, Wilmot, is something that hearing people in that context say, I think would be useful. At the same time, the idea of developing mirror life as a weapon is not one that, given how futile it would be to put it back in the box, given the lack of the likelihood that traditional countermeasures wouldn't work, the ability to develop mirror countermeasures and apply them to every potential mirror life once we had it out and replicating, would be so challenging to consider that that talking about this and setting a red line about it as only a potential weapon seems like not the right forum for having all of the conversation. So what I would say is we need to talk about it in the BWC context. It's already being discussed in the BWC context. And that means policymakers in the BWC discussions need to understand this technology. And they also need to understand the differences between mirror life and mirror biology and the enabling technologies. They need to be part of the conversation about setting potential red lines on the technologies towards the development of mirror life. At the same time, it's not only about, it's about broader ecological consequences, and there are a large number of other elements of the international architecture and the UN system, all the way up to the Secretary General, that are dealing with potentially catastrophic consequences. This would certainly be on the order of those other types of discussions around climate, for example, and our environment, ecosystem, and agricultural risks, food security, et cetera. And so it seems like there is a nascent, I will just say nascent consensus that the statements and norms around a moratorium on mirror life should really be discussed at a higher order UN level, potentially at the level of the UN Secretary General, one of the committees directly underneath of him, and that it should be an all-encompassing conversation. But at the same time, I personally believe that having a statement that creating mirror life would be akin to developing a biological weapon, I think that would be useful. And I think there should be some more work done to discuss the pros and cons of that, and really to get the viewpoints of policymakers who are very well steeped in the BWC. Does that make sense?
It certainly does. And just to note that the discussion at the UN level has started in its Science Advisory Committee. So, but reflections from you, James, on the subject?
I think strong plus one to what Beth said. I suppose my takeaway here would be, I think something like the BWC can be one part of an arsenal of approaches to help to address the risks here, but seems very unlikely on its own to be sufficient to solve the problem. I think one of the biggest levers that we probably have for preventing rogue actors, malicious actors, from being able to create mirror life is ensuring that the barriers to being able to do so remain quite high. So there are a relatively small number of actors that could realistically access the level of resources, the $500 million to $1 billion, and the technical talent that would be necessary to solve what would be very difficult scientific problems that really good scientists have tried to work on and have not succeeded on in some cases. So I think that this points to the critical importance of setting the lines between mirror molecules on the one hand, which everyone agrees, I think, are safe and worthy of investigation, and mirror life on the other hand, which is clearly incredibly risky. Setting those boundaries in the right place that will ensure that the benefits of synthetic biology more generally can be realized, but also make sure that the ability to create mirror life stays out of reach of the vast majority of bad actors that may want to do this.
So thank you. Thanks very much for that. So there's a question in the chat that reads as follows, and we've touched on some aspects of it already, but let me just read it to you. Because we've been speaking about public sector. This is a question about the private sector, and it goes as follows. Has there been any engagement with industry on mirror life? A potential concern might be research that is outside the usual funding streams, that has limited visibility, and whether there could be a profit-driven motive for generating mirror life, such as the issue I already touched on, the purpose of bioproduction of reverse chirality biomolecules. So if there are cost savings over chemical synthesis. So that's the question. Who would like to respond to that?
Happy to jump in on this one. I think in my presentation, I pointed to two reasons that people have been interested in creating mirror life. One, and I think this is has probably been the strongest driver, although I'm not totally sure, is scientific curiosity. This is just something that would be very impressive and fundamentally would be a cool science project. The other is the potential ability to use mirror cells to manufacture mirror biomolecules. So at the moment, this is done through chemical synthesis, which can be quite a slow and laborious process and can be quite expensive. In theory, it could be possible to make those molecules more efficiently if you had a mirror cell. And this is how we make many of our natural chirality drugs. We use cells to make them. I think the overarching consideration here, which I think is most important, is that the people who are working most closely on these technologies, the ones that are developing the mirror therapeutics and have these in the clinic, don't see a need for us to go ahead and make mirror life in order for those to be made. So while I think it's plausible that there could be some efficiency savings, it's very, very clear that the potential risks of doing this would be far greater than any potential benefits there. And over time, we should expect that the chemical synthesis approaches that are used to make mirror biomolecules at the moment will get better. We're not going to, very likely not going to plateau where we are now. And there's a sort of irony here that in order to be able to make a mirror cell, very likely those methods will have to improve anyway, such that you could make the quantity of mirror biomolecules and the complexity of the mirror biomolecules that you would need to make the cell, at which point the value of that cell has actually has decreased. So I think while there are potential reasons that one would want to do this, those in industry that I've spoken to who are working on the most relevant things are simply on board with the idea that mirror life should not be created. And in fact, are involved in all of the discussions and thinking through where should we draw the lines, and will certainly continue to be.
Yeah, and I think this is where the engagement to date with the synthetic biology community has been high and needs to just continue to be high, and to have synthetic biologists, have a strong seat at the table for overseeing this. If a scientific advisory process were to unfold as a moratorium is put in place, you just want people to continue to reevaluate the science and continually reinforce that norm, James. So I think that the approach so far has been fantastic, and it just needs to continue and grow. And there needs to be a reinforcing way to continue to have that conversation, because what we found with other dual use, well, in this case, no use, but with other emerging technology risk conversations, is that if you have the discussion, even if you set a norm, if you don't have a way to continually keep people engaged on the norm, it's very, very hard to not only enforce, but also to keep the people who are at the forefront of the technological conversation really focused on it. And that is also the way to keep the researchers and the funders for that research focused on it, which, as you said earlier on, is one of the critical things that we need to do. If we can't access, if the only way to make a mirror cell is to access the quantity of funding that we need to make a mirror cell as a goal, then there are very specific ways that we can oversee the development, the potential to the development of a mirror cell. And that is by having a clear statement from funders, and also tools like the Biofunders Compact, which currently exists and is promoted by the Coalition for Epidemic Preparedness Innovation, NTI, Sentinel Bio, and others. We need many more funders to sign up to that, and this should be a tenet of it. Mirror Life is not something that we will fund.
So, colleagues and friends, we have 5 minutes left, and I'd like us to use the 5 minutes in the following way. I mean, there are lots of questions in the chat, and we will respond to those questions, not here, but after the webinar. But to say that it's very clear that there needs to be greater engagement with the synthetic biology community. There's a conference that's planned for Manchester, where that in fact will happen. There's a need to engage with the global community of scientists. There is one question here that I'd like you, Beth, or James, to answer as part of your closing remarks. A question about which scientists or groups would disagree with us, and why do they disagree with the stance that we're taking. So the engagement has to be, just to get back to the conclusion, the engagement has to be with the science community as a whole, but also with the policymaking community, with the legal fraternity, and a whole range of decision makers that would be part of the community that says that this should not happen, that there should be a moratorium, because if you don't do this, you prevent this from developing, there in fact would be no countermeasures that would be effective. So if you could give us a quick lighting around closing reflections, having heard all the various points being raised, to wrap up today's session. Let me start with you, Beth, and I'll end up with James.
Yeah, maybe I'll just say that there have been a lot of great questions, and I'm sorry we didn't get to all of them. But I do think this question about who disagrees with us is really important. So far, resounding concordance. There's people who have really important questions about what would happen if Mirror Life was created. There are critical questions about countermeasures, including one in the chat. Would the risks of developing countermeasures outweigh their benefit? And would they in fact be futile? These are questions that many reasonable people are asking, and I think at least where I fall on this, and where the science paper and technical report fell, is that the risks of developing, we would not want to encourage people to develop Mirror Life in order to counter Mirror Life, and the development with the need for medical countermeasures would potentially be completely futile, because it would be impossible to put this technology back in the box once it was out and self-replicating. That being said, the whole discussion and debate around this topic is intended to find people who disagree, if they exist, and to understand whether or not there are disagreements. And so far, there's concordance that there should be a moratorium on Mirror Life. Where there is not yet concordance is on at what part of the pathway towards Mirror Life there should be some oversight and guidance. And that work really needs to begin in earnest now. Our lives and our ecosystem may very well depend on it.
James, minute.
All right. Yeah, I think just to say, I think there has been very broad agreement, as Beth pointed to, around the overwhelming importance of ensuring that these risks are not realized and that Mirror Life is not created. There are, of course, people who will have questions over specific parts of the analysis here. I think some of the key things that people would point to are, is this a risk that's too far away for us to really devote resources to it now? And I think to that, I would say, this is an opportunity. We really have a chance here to get ahead of a risk well before it happens, and make sure that it doesn't happen. So I think this is something I would view as a feature of this problem rather than a bug, and I would really encourage other people to think about it this way too. I think one of the critical things in this area that's been different to other analogous scenarios is that the people who previously wanted to create Mirror Life are the ones that have come forward and now called attention to the risks and said that they don't think it should be created. And that, as well as the broad international collaboration and agreement that we've seen on this issue, I think gives me hope that with sufficient resources and dedication and participation from people like those in the audience, we will be able to make real progress here.
So with thanks to Beth and to James and everybody else who's joined us today. Thank you also to the Pandemic Center team for putting this event together, to Bentley, Holt, Michael Mitchell, Leigh Lovgren, Amanda Kogan, Carly Gasker, Andy Ulig, Anne Wang, Alice Im, and Brendan Beng. Stay up to date for future sessions. Use the QR code on screen to access Linktree, and there you can sign up. A recording, colleagues, of today's session will be available on the events page of our website next week, along with other previous webinars. Follow us on social media for updates, and please feel free to reach out with any questions or suggestions. We'd love to hear from you. Thank you very much. We are adjourned. Be safe, be healthy, and stay engaged. With many thanks.